My name is Cornelia Klutsch, and today we will talk a bit about genetics and how biologists can use genetics to study wildlife populations. But before we go into this, I thought I would talk a bit more about myself. As said, I'm a biologist and I work as a research scientist at a biological station. Here's a picture of it in the summer with a little pond and a botanical garden in the front. So I work at a biological station called Nebius-Svanhoft, which is situated really in the most northeastern part of Norway, and a little wedge here between Finland and Russia. So we're very close to the borders of our international neighbors, and we have a lot of collaborations with those neighbors. But just to demonstrate how close, particularly the Russian border is, here on the bottom picture, you see the biological station, Svanhoft, and you can basically walk to a river, which is called the Pacific River. And we will talk today also about the Pacific River. And then on the other side, we have already Russia, and the middle of the river is basically the border between these two countries. And I will talk about this also a bit later. And what we are working on at the station is a lot of things related to climate change. So we are about 400 kilometers north of the Arctic Circle, and climate change is happening up here quite quickly, and we are interested to understand the mechanisms behind it, what is changing and why. And I have listed a few species here. So traditionally, there has been a lot of work with brown bears and other large carnivores, but in recent years, we have expanded our research task to different species and different ecosystems here in the region. And we are working now from everything from mammals to fish to insects and other things. So, but one of the main themes we have, or one of the main tools, is actually genetics. And this is really what I would like to introduce you today a bit more. And I just have summarized this a bit on the slide. So I will, in the beginning, start explaining a bit what actually genetics is and what we're using and why is this important to us, and give you a few definitions that are important to understand and a few concepts so that you better understand then one of the real life examples I will talk about a bit that we are doing right now, the research we are doing right now at the stations. Yes, so I think we just dive right into it. And first of all, it's quite interesting to think a bit about other people and your surroundings. So probably sitting in a classroom or at home with siblings or with your family. And one of the things you can realize is that no person looks like another person, except probably for twins. And you all have different morphological features. You have different eye color, for example, or hair color, or other characteristics that are different between individual persons. So what we are talking biology about is that you have different phenotypes. So there are differences in how you look. But you share these characteristics with your parents. So usually you are a good mix between how your dad and how your mom looks. So probably you share the eye color with one of your parents and the hair color with the other parent, for example. And so in biology, we call this inheritance. So you have inherited, you got some characteristics from either your mom or your dad. So, and finally, you have a specific carrier of this information, and this is the genomes or specific genes that are responsible, or more generally, the DNA that is the carrier of this information. This is why you look different, and that's why we have this variation in the human population and also in wildlife populations. And I will introduce you in the following a bit more to those terms, to what the differences are, and so that you better understand some of these concepts. So we start first off with the graphic of cell. And there are different structures in here, but the main structure I want to point out here is this sort of pink, purplish, yellow ball, which is the cell nucleus. And most of your genetic information is stored in this cell nucleus. And then you have structures in there that look like these X's, for example, and these are called chromosomes. And I will explain a bit more in the following. But basically, these chromosomes are packed, are basically DNA molecules packed into these structures, into these chromosome structures. And then you see here this tail end is DNA. So to explain these concepts a bit better, I thought the first thing you can think about is a book, actually. And in a book, you have different organization levels. You have chapters, you have sentences, you have words. And we start with the lowest level of organization, which is words. Words are made out of letters. You have an alphabet, and strings of those letters form words that have a specific meaning. And very similarly in DNA, which is also called deoxyribonucleic acid, we have an alphabet basically of 4 nucleotides. So DNA is a structure that uses 4 nucleotides, namely cytosine, adenine, thymine, and guanine, that are organized in a string, and also they are complementary to each other. So you have 2 strands, which we call also a double helix. And the first letters of those 4 nucleotides is basically the alphabet that DNA uses. And the sequence, the series, the string of those 4 nucleotides are basically forming words that have a specific meaning. There's a specific information stored in the sequence of those, like exactly like in words, that is a string of letters that has a specific meaning. If you go one organization level up in the book, this means you have a sentence. In this case, genetics is cool. In DNA, and this sentence has a specific meaning again, right? So it has a specific structure, it has a specific syntax. And in the DNA, you have a specific string of those nucleotide letters, A, G, T, C, and so on, that form and these are packed basically in units that we call a gene. So there's a specific length of those nucleotides that are organized in a gene. And this gene can code for proteins and can be responsible for some of those morphological or phenological characteristics we see, those phenotypes that I talked about in the beginning. It is important to realize that we have sometimes typos in a book, in a sentence, and we may change the meaning of the sentence. For example, genetics is coal. So we have one little typo here in the end, which means that the sentence is changed, the meaning of the sentence is changed, and probably it's probably quite nonsense what we are reading. Of course, genetics is coal doesn't make much sense. But we can have similar changes in the sequence of those 4 nucleotides, which we call also mutation. And this can lead to changes then also in phenotypes and how we look, or in certain functions of genes like protein synthesis. So I will talk about mutations in a bit more, but let's just go further into this hierarchy of organization levels. The next higher level in a book is probably chapters, and chapters are larger collections of sentences. And again, very similarly, we have in genetics, we have chromosomes. So I introduced them in the beginning, and these are a larger collection of genes. So on these chromosomes, several genes can be found that are really compactly packed into chromosomes. And then finally, you have the whole book, with all chapters, with all words and sentences. And the whole book contains all the information for the story that really is one unit that really gives you all the information you need to understand the story or the information, the topic that the book is about. And in genetics, the genome is basically the entire collection that contains all the information for all the functions and all the characteristics that are important for your body. And they're organized in sets of chromosomes. And here in this picture, we see actually the human karyotype, that's also called. And for humans, we have 22 chromosomes. And because humans as mammals have a diploid, we have two sets of everything and the karyotype. We have 44 chromosomes, but in addition, we have two sex chromosomes. Those sex chromosomes determine if you're biologically female or male. So in total, we have 46 chromosomes in humans. These sets can vary between species, so other species may have a different number, but in humans it's 46 chromosomes. Okay, coming back one more time to the concept of mutations. So I said that you have changes. So here in this example, I have three sequences, so three strings of nucleotides. And you can see I have highlighted them in different colors here, that you have different combinations of different nucleotides in certain positions. First of all, it's important to realize that these mutations are random. So they happen during cell division when also the DNA material is copied. And sometimes simply mistakes happen and you have random mutations. And this can lead then, because of these combinations of changes, that you have different outcomes. In this case, a red, green, and yellow fish. But could be other things, could be also not so obvious changes in your in how you look, or but more subtle changes in functions in the body. And another thing to realize here is that mutations are not as such good or bad necessarily because they're random, but they can lead to that individuals in a population, in a wildlife population, let's say a fish population, is better equipped to deal with specific environmental conditions like temperature, or in fish, for example, the levels of the salt level that fish can tolerate, for example. And some others with a different combination of these changes may have a lower tolerance to salinity, to these salt levels in the water, for example. So it can vary, the outcomes can vary as well. So with this background, I thought I'd give you more general overview, what we can do with genetics, and then explain a bit what we are doing and how we are using genetics here at the station. So if we start here, this is probably one of the most prominent examples. If you think about genetics, it's forensic applications. So I'm sure that you have seen in TV, probably a crime series where investigators use genetics to find out if a specific person was at a crime scene, for example. And we do something similar actually here with genetics. We have, for example, the reindeer police up here in northern Norway. And for example, the people from the reindeer police came to us to investigate a potential poaching case, so illegal hunting of reindeer. And they wanted to know if a meat piece in a freezer of a specific person was identical to a meat piece that was found rests in the forest. So with genetics, we can determine if these two meat pieces came from the same individual, reindeer individual, or not, and could give them then our assessment if indeed it came from the same individual, which would then mean that it's very likely that this person who had the meat piece in his freezer is responsible for hunting that reindeer illegally. Then we have other applications, for example, medicine. And there, I think you can think about the recent COVID pandemic, A lot of advances we have made to understand this virus goes back to genetic tools that we try to understand the function of the DNA sequence in this virus and what it might do. More closely related to what we are doing here at the station, biodiversity studies are greatly helped by genetics to understand how many species we have, to find new species, but also to look at population genetic applications to understand how much genetic diversity we have in a population, which gives us an indication if this is a healthy population or if, for example, it's a very isolated and small population and we probably have to help this population to survive. Then we have things like functional genetics. There we are interested in understanding literally the task, the function of specific genes, which is related also often to medical approaches. Then we have population conservation genetics. I will talk about this in the next slide. And then we have, for example, breeding programs. And also this I will talk about in our example here from the station. Sometimes we have breeding programs either for agriculture, like cows and sheep and so on, but also for wildlife species when we try to help a population flourish again. And then it's very important that we have genetic information in the background to avoid, for example, crossing or breeding individuals that are very closely related, because one of the goals is to keep a high genetic diversity if possible. And related to this, we can do pedigree studies. We can find out how individuals in a population are related, either humans or wildlife species. And we can find out also more how specific characteristics are inherited. And finally, we have sex identification. This can help us to find out if we have in a wildlife population what number of females and males we, for example, have in a specific population. And I mentioned conservation genetics. So this is also something that we are working on here at the station. But I give you two examples here. For example, the panda had big problems to survive in natural population, and the breeding program was initiated to actually help this species to survive. And on the left-hand side, I have put a picture of a gray wolf. It's a species that has been hunted quite a bit in the past and is now partially reintroduced to certain geographical regions. And also there, genetics can help to monitor the recovery of those populations and reintroduction schemes. So what do we do actually in the lab? So what we do, we go out and collect a sample from, in this case, from a fish specimen, but could be other species, of course. Then we extract DNA. You see the DNA double helix again here. And then the lab, through different processes, we basically read this sequence, the string of DNA with the different letters. So you see the different colors code for different letters. And then if we run several samples, we can compare those strings of DNA and find differences between them. Or some positions, of course, the same. And what we can do then later on is to determine if we have, for example, different species or we have different individuals. And the number of changes or differences between these two sequences, two individuals, determines also how closely they are related, for example. So this is sort of one quick run through what we do. And the beauty of this is we can do this for different species. We can study fish species with this method, but also mammals like brown bear or other species. So it's quite versatile. So we can use this, apply this for different scenarios. And with this, I come to the example that we are working on right now. And I mentioned the Pasvik River in the beginning. And so the Pasvik River is about 150 kilometers long. And on the right-hand side, you have put in a map so that you can learn a bit more about this river. As I mentioned in the beginning, it's a river that is partially shared between Norway and Russia. So the middle of the river is basically the border. But there's also a part of the river here at the bottom and designated with the letters A to C that is only found in Russia. And the letters we have designated to different parts of the river that are separated by hydroelectric dams. This sort of this yellow-orange stripes here. And basically, it's just a communication tool for us to compare these different parts of the river, what's happening in there. And it was sort of a logical distinction that we have parts that are separated by two hydroelectric dams that were built in the 1950s to 1980s. So there's a bit of information here about those dams in the table, so that these get a letter to communicate where we are working, for example. What's important is that Russia and Norway have different management practices for this river. So in the part that is found here, just in Russia, Russia basically doesn't allow much for fishing here and angling. Whereas in this shared part from the Norwegian side, there is quite a bit of recreational fishing going on. But also, the Norwegian side has decided to install a breeding program for brown trout. This is the species we are looking at. Because there are potentially two problems in this river. The first one are these hydroelectric dams that are probably separating the populations from one another. And the other problem is that through this fishing and also the dams are a problem in regards to destruction of spawning areas. So this breeding program was initiated to get the population, to support the population of brown trout. And the breeding program basically releases about 5,000 fish, 5,000 brown trout individuals every year in the zones D to J. Whereas no release of fish specimens happens in zones A to C. So it's an interesting system that you have. That is a shared river between countries that have different rules and laws and different strategies on how they deal with natural resources and wildlife species. And from this, we formulated two main questions. But before I go into this, I just wanted to show here a brown trout. This is the species we are talking about. And up here, you can see one picture of one of those hydroelectric dams. So these are quite big structures that really prevent migration of those fish species. So they're really separated in that river. And with this, the first question was if dams actually separate those populations, and can we see this in the genetics of those individuals? And does the breeding program that is done on the Norwegian side change the genetic composition? Is there other changes introduced by the breeding program? Because of course, if there are changes, then we have to ask ourselves if we can improve the breeding program. And I will talk about this also in a second. So with this introduction, we used genetic tools to study brown trout populations in this river system in all of these zones that are designated with these letters. And on the left-hand side, you have what we call a bar plot of genetic information. And at the bottom, you see again the same letters popping up. And each vertical line is a fish individual, and different colors mean basically that you have different genetic information. What we see, first of all, is that the zones A to C have a different color. This sort of means that they have a different genetic makeup, whereas the zones D to J, so these are the shared zones between Norway and Russia, they have far more blue. So there's something going on in genetics that these zones are differentiated from those other zones. And we think this goes back to the breeding program that so many fish are released, that basically breeding program introduced over several generations, several breeding years, genetic change in this breeding population. And the second question was if dams actually introduce genetic changes. And there we looked a bit closer at those 3 zones that are only found in Russia, because we know there's no release of fish going on. And also there we can see that this population C and B in particular are quite different. Again, the different colors mean that there's a difference in genetics here. So, which means that dams, even adjacent regions in the river like B and C, just separated by one dam, show actually genetic differences. So dams have an influence on the genetics of those brown trout fish. And then we ask ourselves, how is this happening? What is the mechanism behind this maybe? And what we are thinking is happening that in the breeding program, a few specific individuals are selected for breeding, and that you basically create a situation where you have a very small breeding population. And a concept to understand what's happening there is the bottleneck concept. So I have a picture here on the left. So you see a bottle with different balls in there with different colors, like this reddish, this orange, and this green. And of course, this bottle has an opening, but it is very small. It just lets through one ball at a time, which is then captured by this container at the bottom. And if we, for example, say that you just let 10 balls or so through this opening, what can happen is 2 things. So the variation, which is here shown as 3 different colors, these could be also different genetic variations you find in a fish population. And what can happen is that you have a scenario where you lose those red balls or those red genetic variation, which you call also alleles. So you're losing specific genetic information, a genetic variation. And it means also that the proportion, the percentages of these different colors or different genetic variation may also vary in the resulting population. For example, you have here in this example a lot of orange, although there is also more green balls in this bottle. So you may change the proportion of genetic variations. And we think this is sort of what's happening because of the selection of a very few breeding individuals. By accident, you are missing genetic variation that is found in the wild. And because you're releasing then so many individuals over time, the population changes, the genetic makeup of this population changes. And this is one reason why we think that in the zones D to J, they all look very similar. So they're released across these dams, which makes sense because this was one of the ideas why this breeding program was introduced, to counteract the separating effect of those dams. But what you also see that is that they all look very similar to one another. So they all has basically one blue block here in this figure. And they're differentiated from other populations that do not receive any breeding individuals from this breeding facility. In a follow-up study, we were then interested to understand a bit more about the whole ecosystem, and we looked at the tributaries. These are sort of the side arms of the river system to understand, because before we only looked at the main river, and we were just interested do we find actually more genetic diversity in those tributaries, or are they all the same? What we found can be seen in this bar up here. So where we have, again, for example, our 3 main river zones. So we compared the main river zones to those tributaries. And then we have tributary, different tributaries sprinkled in here. And we have also, from the Norwegian-Russian part, zones, the main river zones like G and H and others. And the result was of this comparison that we have in total about 8 to 11 genetic classes. These are the different colors. And first of all, the result was that the tributaries are genetically different from the main river, which was interesting. And then second, that they are also different from one another. So this tributary is very different genetically from this tributary and from those. So these different colors again indicate genetic differences between locations in the river. So there seems to be more genetic variation to be found in the tributaries. And what this could mean, for example, is one question is that we probably should take individuals from those tributaries to improve the breeding program, that we have more genetic variation in the breeding program. And you can think about a bit if the breeding program is a success. What do you think? Well, I think my answer to this would be it's both a success and failure. It depends on what your priorities are. The breeding program has prevented genetic differentiation due to the dams. So this was this blue block. There's no differentiation, strong differentiation between those zones D to J, this shared part between Norway and Russia, where fish are released. So then you could say, okay, the breeding program has actually successfully prevented this. However, the breeding has led to loss of genetic diversity. So this bottleneck effect that I was talking about. And it has led to differentiation between those zones that are only found in Russia, where no fish are released, and this zone shared by Norway, Russia, where Norway releases fish species. And the conclusion is here that we have to think about a bit what we can improve. And one easy fix would be that we have breeding of more fish individuals that include more fish, but probably also from different regions of the fish, like the tributaries. And one of these things has been done. So what has been introduced after we published those results is actually that now the breeding facility is obligated to use more fish individuals to improve the breeding program. So this is also an example how science can inform management policy to improve things for wildlife species. And with this, I thank you for your attention, and I'm looking forward to seeing you in the next webinar. Thank you very much.